lined up for you. And you can see here we have 5 short presentations for our panel at the end here. We're going to be covering a number of different facets of artificial intelligence within research and development, and a particular focus on preclinical drug discovery and development. So we're going to start off with looking at patents, when to patent, what to patent in AI. And that's going to be followed by a talk on digital twins. And then AI and robots, how's that affecting drug discovery? Ways to reduce animal testing. So I think all these AI technologies will have a very big impact on that preclinical scene. And we're going to finish off, of course, with digital cell clones and cell simulations with some new insights on what's happening on that front. So these presentations will be followed by a panel discussion, which will cover these AI topics and their challenges. And that will be moderated by Uma Guthrie Kamakolano, who's the chair of Early Career Scientists at the Science Council at SETI University. And immediately following that panel, we will have our reception, which I imagine everyone's looking forward to after 3 days of presentations and partnering here in San Francisco. So again, thank you all for coming, and now we're going to get started and kick this off with Jeff Giles, who is the partner at Wample, Bond, and Dickinson, with insights into the AI ID landscape. I'll go ahead and load that slide deck up.
Hello, folks. Glad to see everyone. I'm thrilled that this is the first JP Morgan in a while that I haven't got my shoes entirely settled. So glad that certainly the folks in LA need this weather to stop. But I hope things are going well. I've been doing patent law for 35 years. In a bunch of capacities, both at law firms I've been in and out. I also work with a lot of others. And for full disclosure, I work with ThinkingNode as an advisor in a number of big companies. AI and machine learning are kind of the recent trend. And the way the law works, just so you know, it's always behind the technology. By a lot, okay? It's not, by the time things get set up and you work through the system, it takes a while. So I've seen the very beginnings of gene cloning, transgenics, all kinds of things, and this has happened with every new technology. The patent office doesn't get it right, not right away, okay? What happens is that they actually freeze up. And so there's a huge delay. What I'm going to try to cover quickly today, there's a few things that we do know, and there are a bunch that we don't. The other thing I want to touch on is as part of your patent and regulatory advancement with your company, it's really important to keep an eye on the regulatory side of it. And we're getting some guidance. I think last year I mentioned that they were going to put out some guidance. Well, it actually came out about a week ago. Okay, and it's kind of what I thought, but I'll go through it a little bit to kind of give you an idea. The topics here are way deep. I mean, each one of these bullets I have up here, we can talk about for days and argue back and forth. But I think what as the companies using machine learning and life sciences and AI, they have to really be flexible and understand that, us lawyers, we don't know which way it's going to go. Okay, and so we've been in this situation before, and what we're able to do in patents is put a number of different approaches in there. But just maybe, how do I click the slides here? Okay. Um, yeah, that's the introduction. What I'm going to do now is I'm pretty sure everybody's somewhat familiar with patents and have heard the words, but basically what a patent is a patent gives you the right to prevent others from using your stuff. Okay, that it's not a positive right that you have. It's to prevent other people from competing with you. And just the basics of patents are that all the technologies all have the same legal requirements. They're all applied a little bit differently, but overall, the two main things are that your invention has to be novel, new, and it can't be merely a non-obvious version of what's already out there. Okay? And so that means as you're developing your products, you've got to look, and when you get to the point where you realize, hey, we're doing something different here. This hasn't really been done. I haven't seen it. The other thing is that in patent law and patent issuance, there's a time lag, okay, between when a company files a patent and when it becomes public. Okay? And that time lag's 18 months. So basically, we can look in the patent office and we're blind for 18 months. Anything that's younger than 18 months, we don't know what's there. Okay, we might have an idea, or the company may announce it, or we may be able to figure it out. But that's the other thing that happens. And I think the novelty is pretty easy to understand. Non-obviousness is almost a mythical animal, and it varies. But here, I think in machine learning and in AI, the technology is so new that you probably have a pretty good chance of figuring out how you're different than what's out there. For non-obviousness, it basically is kind of a spark. What's your magic sauce? Why does yours work better? And that's what we try to cover. And one of the things we did figure out so far legally is that the AI itself can't be an inventor. Okay, that's been decided by both patent office and courts. And as always, somebody, the reason why they tried to make the AI the inventor was they were trying to change the ownership of the patent. Okay, they didn't want to put another company on. So what they were trying to do is, which is never good, they were trying to pull fast. And basically, the patent office shut them down and said, no, you actually have to have a human involved, and a human has to be an inventor. And this is one of the few things that's actually decided. But in law, when you get kind of weird questions, there's always somebody trying to pull something. Okay, and it kind of makes sense that patents are set forth in the Constitution. It's that old. And so here, when say for example, a university made the first kind of basic invention, and then it got pulled out into the company. A lot of times, what the company's doing is they're trying not to pay more royalties back to the universities. Okay, and it never works. Okay, it just never works, and especially if you work with small startups, the best way to goof them up is to put a red mark on the company like that. Okay, so you got your university, you're trying to cooperate, then you try to goof them over, and then they just become bad partners. And the whole thing, you're going to have to explain this to the venture capitalists, like, why is it this way? And that's usually enough to make the venture capitalists just go, hey, thanks, great to meet you, walk away, because they're busy, and any kind of early stage IP issue is going to be a problem. Any kind of early stage inventorship dispute or ownership dispute is going to be a problem. So that's not something you aspire to. That's something you absolutely can't do. You won't get there to Series A. Sometimes you're able to go back and repair that relationship, but it's very hard. It's best not to do stuff like that. And usually you're talking about, a 10% royalty. You're already from the university. A lot of times they go, say, well, if I sell over $1 billion, I'll give you 11. You can negotiate it out. So, and the university will rarely give, they will license their initial programs and technology, but they will rarely, if ever, give you follow-on stuff. Okay, now there are exceptions, and in Europe, it's very common for a university to say, okay, everything out of this particular lab for 2 years goes to this company. You will never get that deal from Stanford. You never get that deal from UCSD. Okay, yeah. So it's different between Europe and US. The other thing I did want to say is the US is by far the hardest country to get a patent on machine learning and AI. It's much easier in Europe. It's easier. And what happened in the US, the courts took a detour and basically created a problem in this area that every single time we file a patent, we have to figure out why is this particular subject matter patent. The U.S. is the only place that it's asked. And then and so what I brought here today is this is one of the inventions I worked on. I started 5 years ago in the machine learning diagnostic space, and what's important in AI patenting in the U.S. is that a human has to do something either before or after. And what we've had the most success with is basically having the doc read out the results and then make a decision about the patient that allows them to select a particular drug they need or knows that they have to be hospitalized. This particular invention related to lupus. So lupus is an immunologic autoimmune disease. Very nasty disease, but it goes through roller coaster rides of is it active? Is the patient feeling really bad? And then when you get them stabilized on drugs, they basically are fine. But they're not fine, but they're okay. And then at some point when they get stressed out again, it flares up again. And this particular invention took clinical trials data, and this is only possible because in clinical trials now, people are collecting what's called RNA-seq data. It's very accurate. The levels, you can get about 10,000 genes. This was all tried with happy chip data, and it was too noisy. It didn't work. And none of the classifiers in retrospect go back and work on the happy chip data. So really, it's the RNA-seq data. And most people now do collect RNA-seq data in their at least phase 2 trial. So, this particular invention, you can see that you train the model, okay? And that, in this case, all that clinical trials data was from 4 different sites. So part of the machine learning and the human input was to clean that data up, and certainly that's critical. Garbage in, it's garbage out. You've been saying that since one. Anyway, so this model with good data could classify the patients and say, this one's fine, they're stable. This one's gonna flare. This one is flaring, and this one is in really deep trouble. And so typically, in these machine learning, you train the models with a bunch of the data, and then you run your test data through. Okay, and basically you can see down at the end here, treating the patient with a drug based on what category they're in. Okay, that's what in this particular space makes this happen. Okay, without that last step, the patent office would say no way, no how. Okay, and so this is evolving, and certainly some of, I didn't have a good example for simulations, and I hear about digital twins, and we've been doing that for a while, and so the last thing I wanted to touch on is the FDA guidance. Okay, and in this FDA guidance, I mentioned last year when I gave this talk that I thought it was going to be like a med device. Okay, so think about a defibrillator that's collecting data, and it starts to get to know the person, right? And so it can distinguish, when they're doing jumping jacks versus having a heart attack. So this is going to apply to products that incorporate AI and machine learning, but it's also going to apply to if you're using AI and machine learning to prove up some of your data in your clinical trial. And it's kind of what I expected, at least because of my experience in med devices. Public comments are going to come on this. Guaranteed the patent office is going to change it. Guaranteed they're not going to understand it the first time. And in the U.S., the way the legal system works, the patent office is not last. The patent court has to make the final decision, potentially the Supreme Court, but not usually. So this is something that all our early stage companies trying to launch a product are going to have to deal with. It's pretty complicated, but probably 10 years ago, with software that was introduced into medical devices, they got a set of rules, though. And this is very close to the same thing. And you can think about it, automatic defibrillator, right? The thing's got to go off by itself. Okay? If you think about the diagnostic part, it doesn't have to go off by itself because the doctor will look at it at the end. All right, I'm going to stop there. I'll just say this is, I've condensed days to goofing around. I mean, each one of these bullet points takes so much work. So I think I'll stop now, but certainly this exciting field that's developing, and I think I work with my group does both biology and computer. And that's pure luck. I'm one of those weird guys that's got a PhD in molecular biology, but also wrote all the lab software in my lab when I was there. And so I really enjoy this. It's a challenge because it's synthesizing a lot of things. And I work a lot with a lot of companies coming out of Switzerland right now. And AIDBio Farm is going to be here, but other companies in Switzerland are very, very all over AI and machine learning. So a lot of those companies are coming out of Basel. So anyway, thank you.
And now we're going to have Chris Shaw, the CMO of NTT. Can we just slides up there?
Slides.
Yes, they're here. I have to load them up.
Microphones. He's good. Okay. Hello, everyone. Thanks for having me here. I'm going to go down a little bit different route in terms of AI. What we're working on AI is going to be on the back end, so the front end of the digital twin we're working on. We want to build a population. We're not going to use AI to build a population. We'd rather have the population built. We'll then use AI for the simulations and testing. I'm also going to show you the device that we have right here that will probably be interesting. So I'm from NTT Research. I'm the CMO, the Chief Marketing Officer. I know in this world, some people might think Chief Medical Officer, so I just want to make that clear.
NTT Research is a fundamental research lab, kind of aligned something like a Bell. We're based in Sunnyvale, we're part of NTT Corporation out of Japan. And we have 3 labs at NTT. We've got our Phi lab, which is physics, and they're working on quantum computing and photonics. Our Sys lab working on new cryptography, and our main lab, which is medical informatics. That's what I'm going to talk about today. And the work there is on a pilot. So something I'm going to show you, I'm going to show you a short video that will explain this, and this is what we refer to as it's an ACE system, it's an autonomous force lift intervention system for cardiac care. So it's a cardiac co-pilot. The defibrillator was mentioned earlier, the idea of having to know if it's automated, it's first the doc there. This is similar situations. Patient is brought in having a myocardial infarction. This is a system developed to treat a heart attack as it's happening. As you probably know, heart attack, ischemic heart disease, number one in the world. This is why we went after this. We will. Sorry. Sort of like that. You hear anything? Okay, I can explain what's happening. So I'm sort of told that story, right? The patient comes in, they're hooked up in a system. This is what is this? It's an ACE system, explosive intervention. The doctor sets the parameters, and then it begins to administer essentially a drug treatment for a heart attack. That's what happens when a patient comes in. You have to give the drug, and try to stabilize the heart and get the patient in a stable condition. That usually takes, it can take anywhere from an hour to 6 hours. This is the system, not unlike a wave, right? This is automated. So you have to link up the doctor, you're setting the parameters that they want, and then the system itself will do the work. Okay. We just have the patient's digital representation.
Vascular biology is based on a mechanism
treatment. We can try mediums of treatment. We can find out the best medium. Then we can apply the treatment to physical patients.
So it uses electrical circuit analogs to represent the contraction of the heart and the flow of blood throughout the body. It also has other layers to represent other body systems as they interact with the cardiovascular system.
The autonomous closed-loop intervention system will be flexible enough to not only accommodate new drug treatments, but also mechanical support devices.
There are some patients who have very severe disease. For those severe patients, medical circulatory support devices may become necessary. Jesus can play a role in setting the proper combinations for these medical devices. So the best combinations of drug therapy and device therapies is possible.
A clinician only has to input the desired result from the drug therapy.
One special thing about ACEs is that it will give the desired cardiac output that the physician thinks the patient needs. You can actually type that in the keyboard, and this ACEs system will do it.
NTT Research is working with a number of partners to help bring ACEs through the next steps.
I think the great thing about our system is the clinical feasibility. We can smoothly translate our behavior into the clinical practice.
We're already working through some of the preclinical studies and have partners to help us with that process.
Our main research collaborator through the Joint Research Agreement for development of ACEs and for cardiovascular development is National Cardiovascular Center in Osaka.
ACDC has been developing a physiological and mathematical model of the cardiovascular system and developing the prototype of ACEs.
They had a dream or an idea for ACEs starting since about 20 years ago.
What I actually did last time was the development of automatic treatment system, the treatment cardiac function. Advanced technologies in data science, control engineering, or entity research are indispensable. So by complementing the strengths of the two institutions, I can imagine the future of ACEs. And that is what entity research at NCBC is also about.
Once in clinical situations, the long-term benefits of the ACEs for patients and the medical community are clear.
Not only is it gonna help the clinician in terms of their decision support, but it's gonna directly benefit the patient.
When a patient comes into the clinic with, say, a myocardial infarction, part of the heart tissue is dying, and the longer it takes for you to stabilize your heart, the more tissue could be lost. If you can deliver drugs more rapidly and get to exactly where you need the patient to be, you save that heart tissue and given the patient her best chance of recovery.
ACEs may potentially support the expert cardiologists and clinicians in managing hemodynamic unstable patients. And not only in hospital, but also in a variety of environments, including disaster sites.
The people in the remote area who do not have a good access to the advanced treatment or specialized doctor, but still they can have a better treatment.
We need ACEs because the cost of healthcare currently is not sustainable. With rising costs, and those people who are not able to afford healthcare will get less quality care. And so they'll tend to have this kind of health inequity. We have an aging population, yet we have increasing costs of healthcare. That's not sustainable. So you need some kind of technology that can reduce those costs.
The hope is to get ACEs machines in clinical situations around the world. In turn, the cardiovascular biodigital tools will continue to refine themselves with data from more and more patients as they receive ongoing care.
That digital twin is a dynamic part of the patient's medical record. So it ages and evolves with her, and it learns from each new piece of data that gets collected during a follow-up or hospital visit. A non-common conclusion was found in your left artery and currently left untreated, but your wife is currently stable on standard medical therapy and being free.
And our liability markers of infarction are falling, simply from that.
She's gonna be okay.
The ACEs provide the highest quality treatment for anyone, everywhere, anytime.
If we want to improve healthcare, we need to address all these chronic conditions that keep bringing patients back to the hospital. And we think that the autonomous protein intervention system, ACEs, will be that kind of technology.
At NTT Research, we're not here to just change the world. We're here to upgrade reality.
So you can see, I think we are at, what we're here talking about AI, what's happening in research, devices, drug discovery, all of that. We're on the precipice of major changes in this. I think what's really interesting about we started this program 5 years ago. It was not an AI-based program. AI is now becoming part of it. So we are seeing things move faster and faster. And I think one of the things talking about patents, I'm sure we're going to hear about new devices, new drug discovery. When it comes down to like the hope of AI, I mean, there are a lot of things we talk about here. What it really comes down to is access. There's not enough doctors, clinicians, or healthcare workers in the world to take care of what we need. So we need these kinds of devices, this kind of technology, across the board. So it is a very exciting time. And I think, glad to be here to show you what works. I think what you can see up here where it says, animal models in here. I was just in Japan. We saw this proof out. This works. We did experiments and rested. We had a patient test. We did the test 3 times. Within 20 minutes, we had stabilized the patient. This was an animal test, as you can expect in preclinical trials. But it's something that's working, and the way it's built, we can start with, it doesn't even have to be the digital twin to start, because this forms the digital twin, as we mentioned. The digital twin. And as I said, as we build up our digital population, we will then start to use AI to increase it. We will not use AI to build the population. It's too much of a black box to do that. We won't, in terms of something like this, we need to do that. So that's pretty much all I wanted to go through. These are the 2 areas where we're going with the digital twin. There's in vitro, the one I just showed you, that's the bio-digital twin, and then bio-digital twin on the chip. That's it for me. Any questions? I apologize for the sound issue. Okay, thank you.
Thank you for that presentation, and sorry for the sound issues. Our next presenter is Quentin Perron. He's the co-founder and CSO at Ecos.
Hi everyone. So basically everything here is in Sligo. At Ecos, we are doing generative AI for chemistry, automated synthesis, and innovative biology. We are 75 people based, most of the team in Paris. We have a team in Japan and in US. We are a team composed of chemists, on-chem, medicinal chemists, experts of the pharma industry. So small pictures to try to make you understand what we are doing basically. So 100 years ago, this is how we used to make car. This is Mercedes-Benz, factory, and this is now how we make car. This is Tesla factory. This is a Marie Curie lab. This is how we used to make chemistry 100 years ago, and this is how we do chemistry today. More or less, nothing evolved so much. Still random water flask, separation funnel. Aaron Mayers. So this comparison is not very fair actually, because in the car industry you produce the same car every day, which is actually not the case in drug discovery, because to discover new molecules you produce different molecules every day. I think a fair comparison would be to compare chemistry to gourmet cooking. You try to make very nice dishes, different every day. And can we automatize that? Do you think it's possible to automatize what a 3-star Michelin chef is doing every day? In that case, you start to think that, this might be very difficult. Because you need to think about a technology which is having the creativity of human, and then the ability to make it work in real life. But actually, this is what we have done for chemistry. We are the only company in the world being able to integrate Gen AI with robots to make molecules on a daily basis. It starts here with generative AI. Then once your AI imaging new optimal molecules, you have another AI which enter into the game, which is called Spire, which trying to find for each of those molecules the recipe to make it. It goes retrosynthesis. And then let's say that you have 300 molecules you want to make. How do you organize yourself? What to buy, when, what to launch as a reaction, when, how. And this is what Elaka is doing. And then we plug those AI and those technology to our robots to make the molecule in the lab, and then purification, analysis, and tests. So why did we decide to build such a platform? Is just to improve those metrics. If you have a look to the metrics here, it takes a lot of time to find molecules going from the target to the preclinical candidate. It costs a lot, the success rate is really low. So, I think it is time to go to robots like the car industry has been able to do with the chemistry right now. And the goal of our platform is to multiply by 2 or divide by 2 the speed, multiply by 2, sorry, the speed, or divide by 2 the time to produce those type of candidates. So again, our core solution, MyCare, it's the generative AI which is find molecules visible on the robot, inspire, find the roots, like that, organize your work in the robots. So we're going to go through those technology one by one. So MyCare is not what is people used to do in the past, which is virtual screening. You try to open a lock and you will test all the keys you have. Here with such technology, you design the keys on purpose. So it's called GenAI contrary to virtual screening. The special aspect of our technology that has been trained on organic reaction. So this algorithm has been able to learn chemistry basically. And thanks to this knowledge, you can cover a very large chemical space, 10 to the power of 27 possible molecules which are feasible. This type of GenAI are not random generation of molecules. They are guided by fitness function, which can be 3D information of the protein, for instance, but other models like solubility, stability, etc. I think it's very important, not only for patent, but also in real life, that it has to be guided by humans. So at some point, human, which is usually the chemist, takes decision and guides the generative AI. Like you would do with ChatGPT, you have to transit. And of course, what we generate is compatible with robots. So just to give you a quick overview of this technology, we have different AI models which speak to each other. One predicts ingredients, the other one predicts how to mix those ingredients together. And by combining those AI, you virtually synthesize molecules. So how does it look like? Basically, the chemist can use MyCare and say, okay, I have 2 vector, 1 bromine, 1 nitrogen. On this bromine, I'm looking for something like this or this. For instance, here I want to do Suzuki, put propylene. I want to see aromatic ring. I don't want to see aliphatic chain. In this position, I want to do amide coupling, aliphatic ring, 1 spiral, for instance, no aromatic. And this is very easy to set up that into MyCare, and rapidly MyCare will try to find optimal molecules maximizing those constraints. Of course, on top of that, you want to have a nice 3D good annual profile. Another example may be more complex, where you want to do cycle addition type of things. MyCare knows 2,000 name reactions, so we cover all the chemistry which has been published in patents. You can also do macrocycle. It's a very famous class of molecules with very nice properties and as well very easy to do into MyCare. And as you can see, we have a very nice interface. So for the chemist without no coding skills, you can generate very nice structures. Then you have Spire, trained on the same database, but Spire is a different purpose. You have a molecule that predicts all the routes, all the recipes to make that molecule. And automatically detect the best routes, and you can force Spire to be compatible with your robotic platform. So, for instance, here, this is what we call name reaction. So you can say to Spire, okay, my robot, I master this and this and this type of recipes. Can you find routes passing by those type of recipes? If you have a molecule which is feasible but cannot be made passing by the chemistry you have developed on your platform, it's useless. You can also ask Spire to find routes passing by certain type of partners, price of the building blocks. You can also force the system to pass by certain type of intermediates or bonds you want to break, bonds you want to break. It's available either to score millions of compounds, 1,000, or 1 or 2 molecules, depending on your needs. We call that Spire API if you have plenty of molecules you want to score. Just to let you know, now our algorithm has been selected by Elsevier, so the backend of Elsevier and the AI they have to perform retrosynthesis is powered by ECOS. Okay, so now you have very nice molecules, you have very nice recipes to make those molecules. How do you make it to the lab? So for that, we have developed another interface which is called ILACA, which bridge the gap between a list of molecules and the robots. And also with ILACA, you can develop new chemistry on your platform because chemists, they are not coding person. And you need to make their life easy if you want chemists to use robots. This is the goal of ILACA. So basically, you have plenty of molecules you want to make. Let's say you have, again, to take the analogy of cooking, you have 300 different types of cakes you want to make. How do you organize yourself? What to buy, when, how to mutualize things. And this is done automatically with ILACA. You have 100 of molecules you want to make. For each of those molecules, Spire in backend using Spire API will give you plenty of routes for each of those molecules. And then ILACA will select automatically which routes are the best to be combined together to make sure you use the platform full speed. And the goal is to obtain one route per molecule. And of course, now we need the chemist to validate this strategy because this strategy is selected to operate the platform full steam. But maybe at some point, chemists cannot be happy with one of those routes. So it needs to give its perspective on that, and we offer that possibility into ILACA. So basically, we show all the parameters of a given reaction, which has been predicted by the AI, selected by the AI, and the chemist can modify some part of it, can reject or approve this reaction. Once it has approved it, automatically triggers compound quotation and compound ordering. Once derived to the lab, it goes to this Hamilton, which is like a big fridge with robots inside to distribute the vials and the starting material to the chemist. And then it updates our inventory. Okay, so now everything is set up, molecules, routes, reactions. We reset the compounds and we are ready to launch the chemistry. So just for your information, our robot is able to run 100 reactions in parallel. So for your information, the chemist can do 2 to 3 reactions a day. So one robot is equivalent to 30 chemists. Right now, we master 50 name reactions, so 50 different types of recipes to make molecules. It covers a very large spectrum of organic chemistry. Several papers show that chemists use most of the time 12 name reactions, so 50 is fairly enough right now, but it's a moving target. We're developing new name reaction every week, every month. Okay, so this is the platform we have. It's a ChemSpeed, provided by ChemSpeed. It's a company based in Switzerland. It's a very nice technology, very efficient. So basically, you have reactors where you can heat, mix, and you have also heat or cool. And then you have places where you can put different types of solvents. Here you can put your starting material. Here you can filter. You can add borders, so everything is possible. Basically, basic things you need to do for organic chemistry. So let's say that you have a project 1, 2, 3. For each of those projects, you have different type of chemistry, different type of scaffolds, and you have used Micah to generate plenty of interesting molecules in different positions of those molecules using different type of chemistry. Again, this goes to ILACA, and ILACA will tell you, okay, in the first reactor, you can put 10 bromo Suzuki, 28 bromo copal, 10% to alkylation, organized like this, because all those reactions share similar temperatures, similar duration. And on the second reactor, you can make those one because it's run at room temperature during 16 hours. So this is done automatically, everything is organized automatically. And then, we just tell to the chemist, this is the list of the starting material of the intermediate you just need to put on the ChemSpeed, and then you just need to press play. So the chemist arrive, puts everything in it on the platform, and the platform is just checking all the vials, making sure that there is no mistakes, and just memorize the position, the barcode, and everything. Put this information to the database. The database has already been already been filled by ELACA with all the temperature, the condition, etc. And then the robot is launched, and all this information is then stored in our ELN, which is Matrix. We have applied this full pipeline, Micah, Spire, ELACA, and the robot for in-house program on PKMYT1, which is a kinase target of the Wii family. It's a very well-validated targets. A lot of people are working on this one, especially repair therapeutics followed by various and Atlas in silico medicine. I think repair is the first one with very special scaffold, and everybody basically after repair are doing me-too compounds, so just modifying some branches of that molecules. So we wanted to use this use case to show how different we can be, and using this platform, in less than 3 months, using Micah and our robots, we have been able to identify 4 novel hits with a completely different scaffold. And one of those hits is 20 nanomolar. So in a very short period of time, we were able to identify very active hits with a totally novel scaffold. So I think as a take-home message, I would say that whatever the AI you can have to predict molecules, we see a lot of companies saying, I have the best AI of the world to predict affinity or whatever. At the end of the day, you still need to design molecules and you still need to make it. And this is the purpose of Ectos Robotic Platform, which brings this unique solution to be able to design totally novel compounds at the present speed thanks to our technology stack. Thank you very much.
Thank you very much. That was a great glimpse into the future of drug discovery. Now I'd like to invite Bertrand Ducret to the podium here. He is the CEO of AIDBio Farm.
Hi everybody, I'm pleased to present the AIDBio Farm and what AIDBio Farm is doing in order to accelerate drug development and accelerating the drug development can work on all the value chain. But what I will present today is really on the initial step, specifically linked with animal testing and the new AIDBio Farm next few years we will reduce by 2 the number of animals used in preclinical development. Showing a few examples where we are already doing that. Very nice data we can get. AIDBio Farm is a privately owned company. It's a Swiss-based company, and we are really independent. We are driven by science, and for us, innovation is very important. So we are very all the pipeline is based on innovative drugs, and the business model of the AIDBio Farm is to be really in the development. Core activity is the development. We are not conducting basic research. We are not on the market. So we work with partners for the research. We work with partners for the commercial activity, but we are doing everything which is in the middle. So the track record, we were able to develop 2 blockbusters, triptorelin and oxaliplatin, and we have currently a very interesting pipeline in oncology, having specifically in the core column, it's really the new approach. We invest a lot in ADC, radioligand conjugate, and we are working a lot on small molecule, specifically in the DDR field. And for that, for all the programs in the pipeline, we really apply the new technology, the new approach in order to to make it faster and better. So in order to know what are the new emerging technology, we decided to build an investment fund. We have an investment fund of 150 million. This is an evergreen fund, and we invest in Series A and seed. We are currently 28 companies we invested in, and we are invested, of course, in a company you know, because the company I was presenting just before is one of our investments. So in the investment, we are really thinking of all the value chain of the development, moving from drug discovery, preclinical development, clinical development. And for that, we are really investing in company having a data platform, biomarker, and in silico model. So I think it's difficult to think out of the box by yourself. You can think better out of the box, and the new player, the new starting coming with new fresh ideas. So in order to specifically to make it to speed up and to use less animals in preclinical development, we apply really the 3R principle, which is to replace, reduce, and refine everything around animal testing. And for that, we are supported by regulatory agencies, both FDA with the Modernization Act and several initiatives from EMEA were allowing us to apply new guidelines, allowing us to use less animal testing, and I will show you a few examples about that. Of course, there's still a challenge because in the developers they are used to apply a specific approach. It's not always possible to avoid in vivo study. In some situations, in vivo study are really mandatory. And this new technology are often time consuming. It's expensive, so it's not always easy to apply. So I will just show you how we apply that in the translational research, in the translational medicine. Giving an example in toxicology, not going in the detail, but here on your left, you have 12 different primary in vitro models. And for that, with 148 biomarkers, and we apply a product, and we have a specific data with a product, and we can compare another product, and comparing a series of products of a product plus, could be a competitor, we can see some differences. And so we can understand more on the differences. This part here, you see several differences between our compound and the competitor. And you can see more in detail what does it mean. So we can precise on specific in vitro model exactly what does it mean, this kind of difference, and we can play with reference compounds, allowing to understand what kind of potential toxicity we can mix. So it doesn't replace the tox data, but it can help really to identify what could be the right model, what we can really check in the development, and even we could take a decision. It's not a good program, it's not a good molecule, let's stop it. Another example of helping us to reduce the number of animals. Here we were looking for a combination product using one program of the pipeline and thinking which is a AIDBio 0123 and looking what could be the best combination we can find with this product. So we played with the cell line in silico study, and for that we have some idea of what could be the best combination we can get. We confirm that on the cell line in vitro study, and it's what we show in silico. We can confirm on the cell line, and we validate that in vivo. In that case, we find that the best combination with AIDBio 0123 is cabozantinib. So I think it was very in order to avoid to conduct many in vivo trials, we can really specify and just confirm in the lax in vivo trial what we can see. Another way to use less animal, it's it's a pharmacology study where we want to have a study when we compare one ADC plus another small molecule and to see what if we have some synergy. We can apply either the classical method, which is a PDX method, but we can use the mini PDX, which is a new method allowing to have few animals, just 3 animals per groups, and to have the information in 7 to 10 days. If we compare to use the technology you use usually, it takes 1 month, and we need from 8 to 10 animals per group. So it has a strong impact, and you see the kind of data we get is quite similar. So exploring new indication, new combination, and identifying biomarker. We are doing that on digital cell clone, and we are conducting several in silico study in order to define what could be the best indication for a DDR program which is in development using this technology. And it was impressive because we were able to identify new indication we were not aware before that it could be an interesting indication for us. And we were currently running several in silico study in order to identify what could be the right biomarker, what could be the right combination with our product using this approach. Another way to find biomarker is in that case we can use ex vivo organoid. We can study mRNA coming from this ex vivo organoid, compare them with literature mRNA, compare them with mRNA coming from in vivo animal trials, make a model, and confirm this model in clinical study, and even enrich this model with mRNA coming from preclinical study. Everything can be used together. There is no frontier between in silico and clinical study. Each model can enrich the other one. So, in conclusion, currently we have a regulatory framework allowing us to progress and to use really this new approach in order to reduce the use of animal model and to get much more information that we can get in the past. Of course, there is a lot of challenge, but we still constant. We need to have many discussions with regulatory agency, with industry, and finally, really, what is important is really this combination. Just in silico will not solve everything. We need to have in silico, in vivo animal model, clinical trial, and we need to mix the data. And I think we already were showing in our program that we are reducing the number of animals we are using in the preclinical trial, and it can really speed up the development. So thank you for your attention. I answer to the questions or to be part of the panel in the end of this presentation. Thank you.
Thank you. Very nice display of multiple approaches using AI in early stage drug discovery. And now we are going to hear directly from Dr. Kai Pham, who is the CEO of ThinkingNode. We'll switch to a map, one of the bigger challenges of the night, I guess. All right, first quick.
Thank you, Lev. Wow. Thank you, Lev. Thank you for coming. So today I'm going to take a little bit more time to really explain why the AI we have is a little bit different from what you used to hear about AI and how we apply that and to be able to work with AIDBio Farm, amazing company and team, and to see what kind of result we have. So the presentation has first what kind of AI we're doing and why it's different, and how we apply that for drug discovery and drug development. So I'm Kai Pham, founder and CEO of ThinkingNode, and okay. So usually each time that you hear about AI/ML, it's mainly ML, machine learning. And we have amazing result with that, right? So today it's really the era of data-driven AI. And this allows us to have statistical model and be able to predict amazing things. And of course, the last incarnation of it is ChatGPT. So data-driven approach is only one type of approach. Everything you are doing in a second is pattern recognition. Right? There is no reasoning. You recognize a face, you recognize a piece of music. This is what Kahneman talk about system 1, system 2. The system 2 is where you have reasoning. It's painful. It takes more time because you have, think about the consequences. So when you use data, why you process data is to have a statistical model. Then this statistical model is totally useless until you have somebody to analyze the output and be able to abstract a new piece of knowledge. But this new piece of knowledge is still useless until you can integrate that into an existing body of knowledge. Right? If you give to some financial people amazing knowledge about biology, she or he doesn't know what to do with that. But then this body of knowledge is enormous. But one thing you have to observe here, knowledge is actually the most powerful way to compress data. Right? When you say, for example, all the birds are flying except penguins, well, you have to observe quite of data to be able to have this kind of piece of information. This is very important you have in mind because we do not process data. We process knowledge. And there is a very good reason for that. Is because it's much more efficient to work at the knowledge level rather than the data level. So today, machine learning are focusing on data and on the learning. Another thing you should have in mind is that when we learn, we reason, right? We are critical about what we learn. We don't just take everything granted and try to acknowledge and get that. We are very critical. So reasoning actually is the foundation of learning. Now, this body of knowledge is still completely useless. Right? We have library of books. We have a lot of knowledge, but most of us spend years, right, in university and so to study, to acquire this knowledge. So this acquisition, so now you can see on one side data-driven, which are the machine learning you have today, and on the other side, knowledge-driven is the way that we are processing. But as I mentioned, well, nobody can acquire all this knowledge. Right? So we only have a piece of knowledge because we have to connect all the dots. So naturally, we think that machine reasoning should be able to do that, as machine learning does the same thing on the data side. Well, you don't hear much about reasoning AI. And the reason of that is because knowledge is diverse. Based on the knowledge, you need different type of reasoning. So all of us, we reason by analogy, by constraint, by logic, by case, by probabilities, and so on. So the existing reasoning system are mainly monologic, which means they have one reasoning engine that has to process all the knowledge. The reason why they go to that road is because people say, okay, I have the book that represents knowledge, so I have the brain that represents the reasoning engine. So then my reasoning engine was able to use the knowledge. But actually, in reality, you have to first integrate the knowledge into your mind, and then this knowledge become alive. So when you have one reasoning engine, you cannot satisfy all these different type of reasoning. The research that I'm doing for 30 years, so the first version has been used in my first fintech company, has been very successful. And this is the second generation applied for life science. So what is different here? MARS, kind of barbarian acronym for macro connectionist agent reasoning system. The term connectionist is actually the right term to talk about the neural networks, because most of neural networks, CNN, whatever you name them, if you look at in the better way, they have nothing to do with the real neuron, the way the neuron function. So the real technical term is connectionism. Macro-connectionism is, instead of considering one single neuron as a basic entity, I'm considering an assembly of neurons. So this allowed me to have different granularity and complexity that can be processed per node. And each of these nodes is a mini agent. So now, it doesn't matter what kind of knowledge you have, we can have any kind of logic within the same system. So now I'm going to talk about why it's important, because sometimes people say, well, what matters is to predict, even if I don't understand. Well, if you do self-driving car, yes, if you just predict, it's great. But as soon as you want to intervene, you have to understand the causality. So in this case, for example, we have a system that tries to predict polio outbreak. So you can have a bunch of, input data. Then if you have a lot of cases, you can have the correlation model. Then you can do statistical prediction, but you still don't know how to stop it, because all this input data, you have no idea which one really is the cause or how they interact with each other. On the other side, if you have the causal model, then you can understand what's going on and you can predict by deduction. So, a lot of time people talk about hallucination with large language model. It's not a bug, it's a feature, which means that because it's based on statistic, it will continue to hallucinate. It's why now they added agent reasoning to control that. So when you can deduce and you can understand the causality, then now you know how to intervene. And of course, when we did a drug, we really intervene into the system. So more viciously, let's suppose that in your input data, now if we're in biology, it can be, the genetic data, can be mRNA sequencing and so on. In your input data, you can have an element that has nothing to do with the output. In this case, ice cream. Well, in 1949, I love this example because it's really highlighted the difference between correlation and causation. They found a perfect correlation between the sale of ice cream and the polio outbreak. And at that time, even some physicians advised to eat less ice cream. But of course, we know it's not the cause of polio. It's simply because during the summer, people eat more ice cream and people gather more often outside as well. So it's called a confounder. So if you use only if you use only statistical model, you can eat a lot of ice cream because you can have a lot of false positive. Yes. So, of course, you heard about large language model, which is based on machine learning. And this large language model built what is called a pattern recognition foundation model. Right? On the other side, they are using that to develop for the first time a large reasoning model. So I didn't want to go in more detail, but if those who are interested, MIT has published this year a very interesting article from Fedorenko showing that reasoning and language in our brain are completely two different arenas. And she concluded that it's not necessary or sufficient for reasoning. A lot of times we think with words, right? But actually, how many times you are looking for a word to express what you mean? What does that mean? Is that you already did the reasoning. You already have the answer. You just look for how to communicate your thought. So on one side, large language model knows how to talk, but the reasoning is very weak. Again, it's why they have to add the agent reasoning. And then on the other side, large reasoning model know how to reason, but doesn't know how to talk. So it's obvious that the future will be the combination of the two. The characteristics are different, but I'm not going to go to detail there. So now, how we use that for drug research and development. This, all of us know about it. It takes time, it costs a lot of money, and very little chance of success. So, addition to that, finding a biological target, it's even take more time, right? So accelerating any of these things are important. So it's why in 2022, $10 billion have been invested in machine learning for drug discovery. The drug discovery is about the chemistry. It's super important, but it's not enough. It's not enough because what matters at the end of the day is the biological response to the drug. And you can have amazing drug that goes on the amazing target, but what matters is the cellular response is to this drug. So this is what we are solving. We can generate a digital cell clone for any of these space, and I'm talking about clone, not twin, in the sense that we are using a process we call digital cell differentiation that allows us to generate in 1 hour any digital clone. Then you can do simulation with the clone. So how do we do that? To do that, this is, I would say, the classic one that the scientist is working by. The scientists think about pathways, and then based on the pathway, you can develop a drug for a specific target. And most of the time, it doesn't work because it's much more complex than a simple pathway. And worse, it's dynamic, so it's changing all the time. So how people solve that? So today, people are using machine learning, doing an association between the phenotype and between the genotype and the phenotype. Okay. So by doing that, what you can have is the gene association or correlation. But you still don't understand what's going on inside of the cell. And as I mentioned for the ice cream, you can hit a lot of false positive. The last thing is, this is a static model, because when you take the gene expression data, it's a snapshot of the cell at that moment, and that's it. So this is how we use mouse. We use mouse to model the cell through all the biochemical reactions. So our network has more than 150,000 nodes. Each node are biochemical reaction, catalyst, regulator, whatever you name it. And with this, with the reasoning, instead of having just association, we can have interactions through all the biochemical reactions and feedback loop. The other thing that is important is that we take this snapshot of the gene expression, but because the system can infer how things can evolve, we can create a movie from a photo, if I may say. So we introduce the dynamic, And of course, it's just an estimation. It's not exactly what happened, but it gives you so much more information for each protein or molecule inside of the cell. We generate about 50 million additional data points for each clone that we created. So once you have the dynamic, it's when you can talk about causality, because causality doesn't exist without the dynamic. So now we can better target what's going on. So how we build this human cell reasoning foundation model? So it starts with the data, analysis, interpretation, publications, and we get to the knowledge. And we let AI to connect the dots. Okay, so it functions as the way that scientists do, which means that first we learn what is the cell. We are not sitting there and just look at zillions of data. So we teach first the system what is the cell, and then we use data to optimize what the system has understand really about the cell. Next please. So this is a kind of, it's not pseudo-digital stem cell in the sense that now we are using data not to build the model. That's why we use very little data. So it's important for rare disease, for a number of things. We use the data just to differentiate the model, not to build the model. So from there, we can have our digital clones, and then we can simulate the drug impact. So we have to talk about reducing animal. Yeah, absolutely. Our goal is to develop animal-free drug only. The reason is because we all know, right, animal model is not good based on the result, 5-10% success. It costs a lot of money, it takes time, and we are using, it's a light word, I say using, more than 150 million animals per year. So all this use case, we had it with our customers, and the way we position ourselves is we kind of digital CRO, digital lab. So we work with pharma company, and we are in discussion with some wet lab to partner with them to do the simulation. And once you do a wet lab, you can have the result. Sometimes you still don't understand why you have this result. So we can do the simulation to understand what's going on. So we work in cancer and IBD today, but the system just need mRNA sequencing. This is the minimum we need. If you have proteomics, better, metabolomics, better, but we need at least the mRNA sequencing, which means we are limited to diseases where we can have a biopsy. So if we do a project, we do the validation of the model first. So for example, in this case from the paper, we simulate what the paper has in terms of drug response, and we had a 88.3% correct prediction on this drug. Then we work on validating model with GI50, and our clone reproduce the trend that you can observe in the GI50. And in this case, each line you have here is a cell line. So we reproduce the clone for each of these cell lines, and then we simulate the drug on these cell lines, and we observe that it has the same trend in terms of impact on the oncogene and suppressor biomarkers. On the other side, we work on autoimmune disease, IBD, and same thing, we validate the model first. So for example, LPS, right? You all know in wet lab, if you want to have an inflamed macrophage or cell, we increase LPS, so it's like an infection. So then the inflammation increases. So we took a healthy transcriptomics data of the macrophage, and we just increased LPS in our model, and we saw that it's increased 95% of the inflammatory biomarkers. Then we can simulate drug on and off target, and this is different drug. And this is for a Japanese pharma company. We were working on identifying a new target for people who are resistant to anti-TNF in IBD. So first of all, to validate the model, they sent to us 60 known targets for IBD, and the system recognized 93% of them. Then we were able to identify the novel target by simulating the drug, because we didn't have the data after treatment. So we simulate the drug, and we compare the clone that responds to the clone that do not respond. Then we have a list of top proteins, and this list of top proteins, we knock down one by one each of these proteins to find out which one has an impact really. So an example of how we can use that to evaluate efficacy. So in this case, it's mainly for the validation from a publication. As you can see, we can work on a very small data set, just 10 patients. Because again, we do not use the data to build a model. We use the data to differentiate the model. So in this paper, they have anti-TNF, and they observe that 5 patients partially respond and 5 patients fully respond. So we took the gene expression data of this article, and then we generate outcome, and we had a, in this case, really 100% match. This next. So now, how do we do that really in terms of process for the efficacy of the drug? So we take the mRNA sequencing of the 6 cell. From there, we generate the baseline, the sick clone. Then we take exactly the same transcriptomics data, but now we simulate the drug, and then we generate the treated clone. Once we have these 2 clones, then we just need to compare them and see the impact on the biomarkers. Now, how do we do for safety? Most of the time when people work on toxicity, they work on the ligand-induced toxicity, which means that they are looking for the byproduct of the drug as a toxic element. In this case, we are focusing on the target-induced toxicity. So this allows us, for example, we take a healthy liver cell, we generate the healthy clone, then on the other side, we just added the drug simulation, and then we can see the impact of the drug on the healthy one. So target discovery, I'm going to go quick on that. You understand the principle now, right? We have the healthy, the sick, the protein, we compare with the sick one, and then we can have our top proteins. So information expansion, another way to use it. So you use the drug and you scan the drug through different diseases. So this is, for example, how we classify the different type of cancer. Okay, thank you very much. And what I want to conclude is what we bring to the industry is to be able to work directly on the patient biopsy data, even at the preclinical stage. What does that mean? It's a kind of running a virtual clinical trial even before you test on animals. And all the publication, all the transcriptomics data on the different patient and disease, this is our library, because in 1 hour, we can generate this different clone. So then we can already simulate your drug even before you have to try on any animal. Thank you very much. And I think it's Panam now.
I know. I think I have all that in the clear lab. I still. That is what. model like this. Generally, with other minority. So we can one of those lines. Shoot bunch ago. Then that was like 5 back, and so we lost our. Then we can try to do it. So we ran through all kinds of like simulations, complex models that compare to so very similar to this, but not obviously self-model. Ramesh? Yes. I do a good point, right? Yeah. Yeah. You show. You work together? Yeah. So what are you doing? IBM or? No, IBM I left 8 years back, 9 years back. I'm doing, I've done 2 other startups after that. Yeah. So now I'm advising the startups. Some of them are very seed stage and 2 of them are relatively on a good trajectory. So I think. I've been there with you. I'm running a company on healthcare data. And lots more to do with therapy, doctor. Getting some funding from the government as well? Some funding, yeah. We're funded by the NIH for one of our projects, but that's still looking at only more trauma care kind of thing, but not oncology. That's great. In the past, we had oncology. We were talking about 5 years back. Yeah, yeah. We're also working with. Drink. What happened? Jobs panel. Yeah, we're going to go meet some people. We'll leave at 6. Jonathan, we have to meet the Swiss people at 6. I wanted to meet the CEO of weekly. Okay, CEO first. There we are. Have you been back to the IBM folks? Not really. I have to get back. One side. Both funny. You work with research or have you research or? Not right. Not nobody now. When you were at. Oh, I will. So you know Jim Spoorer? Yeah. You all met?
Hello.
You know, I'm talking
Good
about
evening,
fairly
everyone.
regularly. All right.
For your patience,
I'm
we got a
part
part.
of his, Isaac,
Today,
one of the ambassadors.
let me introduce myself.
This
I'm
one.
Murali Kaplan, chair of early career scientist with City Institute. And I'm a medicinal chemist and also
Bio.
Today I'll be joining this panel. I'll be asking questions mostly. So let me introduce the topic. In this era of AI ML digital advancements, there are new ways of drug design and discovery. So we'll see what our panelists have their suggestions and listen to the suggestions. And you can ask questions as well. I will start with the first question. So, what is the current state of AI in drug design and discovery? While they answer the questions, they'll also introduce the panelists, will introduce themselves. So I'll pass you the mic now. Please introduce your names and you can, each one of you can share your suggestions about the state of AI in the drug design and discovery.
Yeah, hello, nice to meet you all. Thanks for coming. I'm Ben Nugans, CMO of Target Health. We are a CRO. So my background is in neurology. I trained at Harvard and in biomedical athletics. worked half of my life in renewable data on the academic side and renewable data on industry, focus on drug discovery and drug development, but more on the clinical side with the human eye on data, the benefit for the patient. So some of the questions might be a little too premature for me, but I'll try to respond from the clinical side and what is then also very important for the regulatory purposes, and that might be something that we can would also focus on, because that is something that we see frequently when we then come and deal with the data that very innovative companies bring to us and then struggle to catch. But yeah, I'll give it to Tom.
Sure. Thank you to Guy and the team for inviting me. So, always have an accent. I'm in Australia. So, I sit on the investment committee for venture fund. Investing in, I guess, the later stage, but drugs and platform as well. I'd say my day job is I work as a deputy research at London. We do research trials in drug study devices, digital AI engineering, and obviously on the investment side, interested in looking at investment of different platform in drug discovery and development. So strong interest in AI medicine. I created a book of AI medicine 3 years ago, looking at different aspects of AI in European medicine. That's including drug discovery. I just pause there and then maybe can comment on, I guess, the landscape and what I see is obviously from Australia, but also in the region, certainly in Asia. And when you look at some of the recent reports coming out, some of our previous speakers, including Colin, mentioned about some of the big farmers testing in-house AI-powered drug discovery. So, for example, check or Institute, something like 632, a few others that actually investing heavily as well. Certainly, I think as some of the previous speakers presented very well, that you can actually build it, but what does it mean? Can you make it? And can you actually prove it? And there are some other experts that perhaps expand on that. But ultimately, it's all about de-risking the whole process. And I sit on the other hand, when we run clinical trials, and we can touch on a little bit about the ethics and so on and so forth. For example, if a drug is been designed in silico, no animal testing, nothing, you have to actually disclose that to the patient. This has not been tested on animal. There's a high risk of failing as well, so it's essentially successful. But that's something that further down the track. So the ethics, the regulatory, as some of our speakers have mentioned, ultimately putting my investment hat on, certainly would be keen to look at why it's actually de-risking. Actually, ultimately, and the question everyone asking is, so why does it have to be a $2 billion, 10 years exercise? Can we actually improve reducing that and get a better return? And some of the speakers here actually do that.
Sounds great. So, what is the role of AI, simulated AI in biology studies, preclinical, non-clinical studies, related animals? That's a strategy. Or will it cut costs? Or what is your view? Is there any timeline reduction that can be observed?
I think so. I'll answer briefly that you're a big fan of lots of expertise here, but certainly I think where we see obviously in the early phase, and I recall FDA announced the Modernization Act 2.0 early last year, and I mean we look at noise, perhaps some chip, so so forth. It's not going to replace any foreseeable future, in my view. Still need sort of the animal models, the toxicology, tox in the pipeline. So to actually, how I see that is AI and drug discovery development will be running in parallel. It gets to the point that, you know, crossover, where I think we can actually get the sort of animal models. Actually, just on that note, just maybe a question of hand, Kai might know the attendees, but who's here actually working on some of the AI drug discovery? Yeah, that's fantastic. Anyone from investment community, anyone in this? I think you're going to be popular. People come and talk to you afterwards. Yeah, I'll just hand up to some of our speakers.
Yeah, so you were talking about, you know, how AI is applied for drug R&D. I guess you understand from my presentation that on one side, people are focusing on the drug using machine learning, and on the other side, to be complete, for example, we don't do anything about the drug, but we're focusing on the cell. And to model the cell, we need a different AI than machine learning, because we want to understand the mechanism of what's going on inside of the cell. So that's why we did the large reasoning model. So I think when you have these two things combined, and then when you have pharma company like AIDBio Pharma that really believe into that and really push for that, then things going to happen. Because we can have the technology, but if the needle is nowhere and the pharma industry don't believe in it, it's not going to go. So I'm super optimistic because of all these elements that are complementary come together today.
So also what's interesting about what's happening right now, I believe you're right. There's the discovery side, and then, okay, how are we going to test this? So whether it's models like you're building, or we're talking about digital twins, but then to throw something else in here that's kind of fascinating is the hallucinations, right? You said it's a feature, not bug. And it's a feature that might give us something novel. So there's opportunity here, even where we don't see it. And I think that's what's exciting about these times, is that we're going to see, you know, new drug discovery, certainly, but how do we test those drugs? And also, we might have discoveries that will happen just as a result of that feature. So we're on the cusp of something, and I think the big thing that I think we need to pay attention to, what's important, is the speed. Right? We are going to see, you know, rapidly changing, rapidly discovered drugs across the board that'll be happening most all as it's always going to be that way. But we're gonna learn faster and we're gonna test faster. And I think that's something that's really exciting.
Maybe I can add to that. I think what has become clear through AI is actually what not just what we know, but also what we don't know. And if we go back like to the animal model, I mean, I don't know who has done animal studies, but I mean, what's your guess? I mean, if you test mice, what gender do you preferably use? With female or male? Okay, who's for female? Raise your hand. Who's for male? That's 3 hands. Okay, but the majority of was for male. Actually, it's female. Why? Because the house then it's a litter of 5. The male just kill each other, and so that's why you primarily want to do female, and also it's handling that you test the white mice, C, black mice, E6, very alcoholic kind of mice, and then you do five mice per group, do the T test, and then I think AI will help us to say that this is not really enough and that we need more data. And I think that's really something you can see in very promising and probably help us a lot more and better to understand biology.
Just to quickly note, I think the speed aspect is quite important. Just listening to Ixel's presentation, when you mentioned about robots could potentially replace 30 chemists, in terms of making ads, isn't it? So it come a cost. So in terms of when it comes to typically in any advancement, you find that it's actually investment in the first place. So there's three professors, economic professor from Harvard spoke about in terms of the implementation of data, where it's actually the foundation of actually investment in the first place. Now Kai working on AI models, and it's in fintech earlier, but it hasn't been picked up by pharma, by biotech, because it's actually not, the return is not straight up. And I think that's where, but what you see is the explosions of the investment in the last 5 years, which is somewhat in a way, look down, if you analyze it further, I would think that some of the pharma, there's a bit of a formula as well. Now, they got money, right? BioNTech got a lot of money from their product, and so they have money to spend. And so do you think an AI company of about 100 people worth, over $630 million? You have to generate a lot of, returns, right? So it comes back to speed is one thing, and then I do think being optimistic as well, where you see all of the possible changes coming, but it will take time. Even though it's a bit like if you build a high speed rail train available, like high speed train, but you haven't got a track to actually run that or sufficient to actually manage it, or people to actually maintain it. And I think it's similarly with drug discovery. We talk to any of my colleagues that possibly still, they don't want to stop them. It's all is still in simulation. On the one hand, I have to do that with others. Well, there's patient digital twins. There's cancer patient digital twins where I think John before bio digital twins, same thing. Patient need to be aware of their family to understand that this is how it's going to change. But it comes back to, I think, that economics. And in my teaching, the models that I use is actually, we tend to focus on these 4 phases. So we're all focusing on, I guess, the speed, time, and its efficiency. But it comes back to the ease of use. And to pull on the data, it's going to be for the patient, for the doctors. So it's the user experience. And then it's the economics. So I mentioned before, economics is hard to actually measure until down the track. And then if it's really, and you're talking about, female, really sort of generalized ability, then it's an equity. If you run a drugs, that actually working on, it could be coming down the trend that if you have pay higher premium, you get a drug that is, have a higher chance. Or so like in healthcare, you pay high premium, you see a real doctor, otherwise you see, the one chip. And then the last thing is really the ethics. All of these, and I think one of the speakers earlier mentioned the FDA regulations and so on. Still coming back to the ethics of implementation. Lots of ways to think about it.
So we have been talking about few cases where patients have to be asked. So what are the ethical considerations that has to be taken care in view of regulatory environment? We have all these regulations to be followed. Are there any global regulations? We have many local regulations. Maybe I can extend the question. So depending on the data available, how do we bridge the gap between the data available and the AI capabilities to, to come up with success predictive drugs that can be more useful, that can help people reduce costs, timelines, effective time management, resource management. Do you have any suggestions?
Personally, I think that it's not AI against biology, it's all together, and I think AI would create new hypotheses. And after you build the hypothesis and you adjust to assess. We experiment when we develop drug. So we experiment, of course, we say we have experimental drug in development. So we see on patient, but before moving to patient, each of us, I think, I don't know if someone in the room would be ready to take a medication which was never tested on animal. I think maybe a guy, you're the only one. But we need, of course, to associate all this technology. And currently, when we have a human or animal, we can extract a lot of information. That's where it's coming, how we process the data, where it's coming to the AI. We can make a lot of stimulation. We can learn a lot. I think for me, really, development is continuum of in silico assessment you can make, in vitro, in vivo, animal, clinical development. And you are always learning and using the new data in order to improve the knowledge you have of the drug.
Sounds great.
Yeah, I just want to talk about sometimes people think about do we have enough data? What is the knowledge gap? There are two types of knowledge gap. The first one is factual, which means that you need more experiment to have the data, to have this particular fact of knowledge. But there is another one that actually is there, but we need to connect the dot. So when you have all your knowledge, a lot of time, we discover new knowledge with existing knowledge, but we don't do the connection because it's too much to do it. So AI in the reasoning part can help to fill new knowledge just by processing knowledge itself. What Bertrand said, absolutely, we need this kind of feedback loop, right, between the web lab and the simulation. And then from the simulation, we maybe think about a more specific web lab to provide factual knowledge that we need and so on. So it's a integration process.
Yeah, so maybe I can add to that. So what Kai just said, connecting the dots, and I would say longitudinally, so like repeated measures, and depending on whether it's animals or patients, across healthcare systems. So longitudinally, but also vertically across proteomics, metabolomics, and so on. So I think the dots vertically to create like a GIS for the human being. And then once you have that data set curated, and then have it analyzed, have it exposed, so that you have different people, scientists, clinicians look at it and analyze it. One example is the mimic data set from the DIC from Beth Israel. That's now in the next iteration, and so many papers have been published. A small curated data set. That's what has to happen to really understand data and really make them usable.
Okay, and I know Kai has introduced us to this simulated AI environment. So are there any other data sets available for the preclinical studies? As of now, we are discussing preclinical, and are there any disease models or simulated environments available for animal models as well? Can you explain on that? And how do we build it further to help our drug design and discovery?
So you're talking about data that because okay, we talk about large language model, and large language model kind of taking everything. But I think for biology, it's a very different type of data and knowledge in the sense that it's not just text, it's a lot of numbers, figures, charts, and so on. So I think it would be important to make kind of specialized set of data that is more biology rather than going after AGI. And prefer to have something super specific expert in biology than having something that can kind of answer everything generally. So I don't know if it's exactly the question you were talking about, but I think this is what we it's kind of, there's so much data, but completely in different places. Can we have, you know, a way to integrate in a biological way all this data so then AI can really process that?
Are there any test instances where the disease models have been tested in simulated way and then taken into the animal models, either in non-clinical or clinical studies?
So can you repeat?
Are there any studies that you have conducted using your simulated environments and then have taken them into the animal models which are successful?
Yeah, so the way we validate the model is each time we start a process, we generate the clone, and then we see if the clone, the model responds to known results. So like I mentioned, LPS. Everybody in the wet lab know that you want to have an inflamed cell, you just increase LPS because then it looks like there is an infection. So it's what we did. We take a healthy gene expression data, we generate a healthy macrophage, and we just increase LPS. And we saw that 95% of the inflammatory biomarkers definitely.
Sounds great. Audience, do you have any questions for our panel today?
So my question is, it's a lot more complicated than you think it is. Let me explain. A human being actually gets exposed to the environment, changing nutrition along his lifespan. Now, when you don't know what those inputs will do in a simple cellular model, you're looking at a really difficult prediction of whether or not a drug's going to work and who's going to work it. And so if you don't have a holistic idea, I'll give you another example. If you take postmortem data, right? Everybody is available in public database, every AI guy is shoving it down an algorithm. But do you know how that patient died? Do you know what happens when you don't have DNR? They starve to death. So when you have a massive calorie deprivation in that data set is a major confound. And now you got a collection, a collection of patients, some that had DNR, some that didn't. The whole nutritional status is completely ambivalent. You don't know. And so if you take this data and in a mathematical model, it's very precise. Mathematicians are very precise people. Axioms and assumptions are a big part of any algorithm. And you stick this in, you will get ego. Garbage in, garbage out. And I think that consideration has never been properly understood in making this happen in clinics. And that poses a serious problem because my thinking is one of your biggest liabilities is legal. Okay? In a business model, you can publish all the data you want in academia and nothing will happen. The minute you make a wrong prediction and you kill a patient, from a business point of view, you're now looking at something very serious like legal liability, just like any pharma drug has. And so my caution is we need to understand all of the variables before you can create a proper interface between AI and all the lovely things. And I think it's going to work, but the idea is do it properly. Right? Thank you.
I couldn't agree more. Who's a lawyer in the room? A few lawyers. When I'm on a panel with a lawyer, I'm happy because the legal people will actually understand that. But what I would say is that the legal framework is always going to be behind the technology advancement. And I don't think anyone on the panel would disagree with you in terms of, I guess, being cautious and you know, as an investor, I see companies coming presenting. I think a few speakers mentioned that as well. That is actually promised, you know, that can, you know, give you a whole world. But if those who actually work on in the AI space, that is actually involved in patient data, looking at patient improvement, improving outcomes and so forth, people understand. I don't think it gets to the point where, even though we're talking about digital twins, it's still a computer simulation and mathematical models. So what you're talking about is life is complex. AI is trying to simplify. And we're trying to, it will not get to the point of crossover. So there are ways that we're trying to, you know, improve it in terms of outcome for patients. But what we all know is it's like, why does it actually cost a few $1 billion over 10, 12 years? We're striving with this thing. Just on data, and I'll be interested to hear from the rest of the panelists as well. A couple of things that we haven't touched on is that in clinical setting, when we run trials at my hospital or any other hospital, is that we don't report negative results. Things don't. Like, absolutely. That's the key. Because in any model, you have a feedback. You can't go one where you know, all the results you see is positive, it works. But the one that don't work is actually quite important. And then secondly, we haven't touched on is synthetic data. Sort of touch on the actual application of synthetic data and how that's actually going to help to improve the models that we look at. So I think I'll stop here and interested to hear from other panelists.
Yeah, I would like to add something. Things are not black or white. What matters is, do we make progress? Do we provide something that is better than what we had? There is about more than 30,000 diseases. I challenge any physician to have that in their mind. So we are imperfect, but still we help people, we develop drugs. So when I talk about the digital clone, it's not really a clone, of course, but it's way, way better than any human can have put his or her head around. So I would not be as, you know, either we have it completely or we don't have it. What matters is, do we have something that allows us to go to the next step? And it's how human we have always go, right? Incrementally through experience that work and work each time user bring better results.
Next question. Sure. Thanks. Nice panel. Very interested to hear, sort of in the 5 to 7 year time frame, where you think in the drug discovery and development pipeline, where AI is going to have the greatest impact on that somewhat shorter time frame, like the 5 to 7 years.
Well, I'm going to preach for my church. It's the expression, right? So I strongly believe that we are going to continue to improve our digital model. And this, as Bertrand mentioned, we can do simulation and reduce the use of animal because it's just not good. Animal models are just not good. So it's not like in one day we're going to replace that. But I think in the next five years or something like that, I think people are going to trust more and more on the digital model to make sure that the model they have from the animal makes sense. Right? If the animal model is positive and the simulation is positive, then you think a good chance. If the animal model is bad and the model is bad, then forget it. But if the animal model is good, but the simulation show it's not good, then maybe this can help you to dig deeper where can be the problem before you decide to go all the way.
I will pray for my yeah, I think biology is very complex. I do not believe that we will reach one day 100% in silico. And anyway, you need to pass by the physical world and to test things. So my perspective on that is, again, I think AI makes a lot of sense as soon as you put it through robots, and I think it's a natural process of things. We're going to see a lot of robots in the years to come, which can help people and a lot of stuff powered by AI inside. So our vision at Ecos is, I think in the futures, the AI will be able to make its own experience. It's all experiments, thinking by itself, imaging new molecules, executing the molecules, closing the loop, and start exploring the chemical space and by itself, actually, without maybe all the bias that human can have. So this is my vision.
Just to complete that, I think look at what we are obliged to do, what the regulatory agency are saying. Please conduct randomized clinical trial. What does it mean, randomized clinical trial? It's really, we don't know. We don't know exactly what is one human person. The best way to compare the treatment of one person is to take a population, to treat the population, and to have a comparator. And it's how it's working. And I think it will work. I think you say in 7 years we will have still the randomized clinical trial. Of course, we can improve. We could create randomized with digital arms, so taking very high quality data, building a comparative arm, and just have one arm, and we can compare to this very high quality data. It's very difficult to have this high quality data, and we have to make a really high quality job in order to convince authority that the product is working. I think for me, I see this kind of improvement in the clinical development. I see that for the we will have better access with AI to the data, and human being is big database, and we can extract a lot of data. So meaning that in the future, maybe we could conduct clinical trial with less people, with that much more data we can extract from one person in clinical trial. That's something we could see in the middle term view. Of course, I think major changes will be preclinical, because preclinical is less regulated, and I think we can apply a lot of technology. But in the end, you will always have to go through clinical trial, and that's the way to be sure we have a safe drug and efficient drug.
Yes, please.
A question for all of you. I'm a great panel. You are modeling just a single cell at the moment, right? Obviously, that's very hard. But looking at also modeling multiple cells at some point, be able to study that. That's one. Second question I have is, would external data from the existing, clinics seeing patients with multiple conditions help you model better your drug design and your drug? So what I'm saying is, can you also use healthcare data, other data sets that can feed into how you model and maybe improve your models from a macro level? So there's a micro level modeling and there's a macro level modeling. So it'd be good to hear your opinions on the panel.
Yeah, so I'm going to answer for the cell. So today we're focusing on the cell or the biopsy. And sometimes people tell us, yeah, but it's the interaction between cells that are interesting. So today we don't have the capacity in terms of resource to do that. We are looking for funding as well. But when you use the gene expression of the cell, the gene expression is already the result of the interactions with the other cell. So we have quite good results, even though we are focusing on one cell or biopsy, because we are focusing on the gene expression, not on the genomics, for example.
I was just add to that. I mean, what I mentioned before about the negative results data, that's one part, but also the real world evidence, how it's actually been, patients on medication or certain drugs, then, all the information will be collected in the real world. That's where it's going to come and fit in. And I mean, do we can wrap up soon?
Yeah, I think we are running out of
Oh,
time.
okay, all right, then I'll just make a comment too. I was going to answer the questions about the 5-7 years time, horizon, because I think that's quite interesting as someone who invests. You always want to look at, sort of in the future. And of course, there's AI and so on and so forth. But it's just a tongue-in-cheek comment. Perhaps maybe what I see that in the next sort of 5-7 years, you see some of the tech company will be sort of heavily involved in the space. Maybe, who knows, NVIDIA could be buying a few pharmaceutical companies and actually running it. And it's kind of, when you think about it, maybe it's a joke, but who knows? I mean, they can put a lot of money, put a lot of power, in terms of computing power. That why not? So I don't know, Kai, whether you're not on NVIDIA doors, to fund your project.
Well, because we don't use GPU at all.
All right.
Because we have the knowledge level and reasoning, so it's much more efficient. So yeah, we don't use GPU.
Thank you. So any more questions before we wrap up? And do you have anything else to say?
I'll just thank you for all coming. And so this is our second edition here. And thanks again to PwC and of course all our sponsors and partners. Thank you for coming to share your experience and so on. So now let's have a drink and chat.
Thank you.